So I'm going to be speaking about the importance of natural history studies in drug development for rare diseases. So let me just let my slides catch up. Okay, here they are. So I'd like to discuss why we need natural history studies and their importance in informing rare disease clinical development, say a little bit about study designs and the concepts that go into natural history studies, and I'd like to give some recent examples of how natural history study played a role in designing and conducting marketing applications or clinical development programs able to support marketing applications. So natural history studies have many functions, such as patient care, and I know that you'll be hearing more about this from speakers a little bit later this afternoon, but I'm going to be talking specifically about how they can help us in developing drug development programs that are able to support marketing approvals. And we believe that the best access for patients to therapeutic interventions is through an approved drug. So the end in mind being we're trying to get to marketing approvals and the data needed to support those. And that is one perspective. But what is the path that we need to follow to try to get there? So in that context, what I'm talking about is natural history study being used to inform drug development. In the United States, to get an approved drug, you need to demonstrate the effectiveness, safety, and favorable risk-benefit of a drug, and that usually requires the conduct and design of adequate and well-controlled studies. Adequate and well-controlled studies exist within the context of the disease. What do we know about the disease? Who are we treating? What are the expected outcomes? And all of that requires a scientific foundation of some kind upon which to build. Drug development takes place in a stepwise process, and we're always trying to learn and inform and move forward. So knowledge of disease natural history for any clinical development program is an essential element. It's often the first element that we look at when we're trying to design these programs. And so for rare diseases, in general anyway, not always, but in general, the natural history is often poorly understood. And what I'm hoping to persuade you of is that this, if we have this knowledge, we have this information, it can help us not only to conduct these programs, but to do them efficiently and in a scientifically sound and rational way. And so what is it that's so different about rare diseases? I think this is probably well known to everybody, but certainly they are rare. Certainly there are a few patients with the disorders. This will limit our opportunity for study, even for replication and for learning. Patients are very, very precious. But because of these factors, a few physicians will have seen these diseases. There are often few experts in the field or treatment centers. So because of these factors, they are poorly understood. And so this is why we need that information. There are also a number of different rare diseases. Sometimes we speak as if rare diseases represents one entity. If I do this for one rare disease, do I do it for all of them? But they are very different. It's very heterogeneous. And since many of these are genetic, phenotypic diversity within the disease is really expected and often not well described. And very importantly, and I'll show you in just a minute, there's often very little precedent for any of these drug development programs, which is certainly in contrast to common disorders, which is another thing that can really help us when we're trying to get to that all-important marketing application. So all of this taken together, it requires more and more careful planning than for non-orphan drugs, and again, we do need that scientific foundation upon which to build. So this is just a snapshot of CDER, Center for Drugs Rare Disease approvals from 2011 to yesterday in 2012. I'm just limited this to new molecular entities and new biologics, since this would represent the first time they are available in the United States. And in that time period, there were 15 rare disease approvals at CDER, which has about 90% of the orphan drugs at FDA. more than half of those did not have disease precedence. We'll put them in a yes and no column. Yes meaning disease precedence, and what we mean by that is they're an approved drug in the United States for that indication already. Has somebody already defined a regulatory path as something that we can use or learn from? And for more than half of the time, the answer was no. And I think if you can see this from the back, but it represents a substantial diversity in the diseases under study. We have rare cancers and genetic disorders and a number of other conditions as well. So contrast that with common diseases, enemy and new biologics approved in that time. There were 24 of those, and only 2 of them did not have a disease precedent. So that is a big difference. So putting this another way, using a construction analogy, if you're trying to build a house, or I guess this looks a little bit more like a monument, but it's you have to dig the foundation first. You have to set the foundation and put the support beams before that you can layer the clinical trials on top of that. And this, again, is a main contrast with common diseases. Common diseases, we often are starting from a much higher place. The support is already there. For rare diseases, we're often starting at the beginning. We're describing the natural history, but also what is the pathophysiology of the disease, the mechanism of action of the intervention that you're evaluating, and what are the expected effects of that intervention. And you'll take that knowledge together and then design your clinical development programs. So these are just some of the things that you would need to describe along the way. Many of them occur in the basic science or in the translational phases, but taken together and building upon these, they then, in a series of steps, will inform later clinical development. So some of the things at the bottom, for example, would be biomarker or clinical outcome, first identification and then development. These can be started early. These can be piloted in natural history, for example, and then be used later on when you're designing those efficacy trials. So once again, just looking down the road, looking for that end point in mind, we're trying to get to the adequate and well-controlled study. And just these are the 7 elements of an adequate and well-controlled trial, but just to highlight a couple of them, and how can natural history help us. One of them is you have to have a comparator. Is there a control? Should it be concurrent or historical? Natural history, the characteristics of the disease, how rapidly progressive, how serious is it? That can be very informative, not just for design issues, but also for ethical issues. Should we have a concurrent or placebo control? Which subjects are you going to select for study out of the population? And very importantly, do we have well-defined and reliable outcome assessment tools? Have they been developed? Are they ready for us when we need them, when we are designing those efficacy trials? So hopefully you can see how this information early on will be available and will feed into programs as we move forward. So then the next question that we usually get is, okay, I want to design that natural history study, so what makes a good natural history study? So, and what exactly is it you mean when you say a natural history study? So, I'd just like to go over a few concepts there. Natural history study and registry are often used interchangeably, but I'm not really talking about the same thing. Registries are a general term, inclusive of many things, and many of them are very useful. For example, you could have a communication registry, or not infrequently for rare diseases, there will be post-marketing requirements after a drug is approved. So that can be done through registries, for example, to further define safety or the effects of the intervention. But natural history, what we're referring to here, it has a very specific purpose. It's intended to describe the disease itself, and it's intended to be comprehensive and as granular as possible. This is a definition of natural history from Steve Groff's book. I think Steve is probably speaking a little bit later. But natural history is the course of the disease from the time it starts through its various phases, right to the end of the disease, where the disease is ended, either patient's cured, disabled, or has died without external intervention. Putting this another way, Institute of Medicine in their rare disease report from 2010, they described natural history studies as pillars of epidemiologic research on rare conditions, but again, the intention is to track the course of the disease, the full course of the disease, over time, and that time component is very important. So are there variables that correlate with the disease and the outcome in the absence of treatment, and what are they? Are there demographic, genetic, environmental factors? You would expect these studies to vary in the fields you're capturing or in the design depending on the disease. Wouldn't expect the same thing for multiple sclerosis as for rare form of cancer, as for chronic kidney disease, for example. But since a lot of these are genetic and mutational defects are important for many diseases, some of the minimum information expect to capture would be genotype information, phenotypes, and particularly the full range within those phenotypes. The course of the disease, very important. Is it chronic progressive? Is it episodic, relapsing, remitting? Is it just one acute episode and then it's over? That will definitely impact on your study design. And very important to identify the major and important morbidities and mortalities and the time course. It can be very different for some of these for multisystemic diseases. And then there will be other elements that are important depending on the disease. But also another important factor is you're trying to consider the larger experience. Pooling multi-center experience, going to more than one treatment center, international if you need to. It's very important to try to describe the full spectrum of the disease to the extent possible. And what we're finding is with very low prevalence disorders, even pooling a small number of patients from different treatment centers can be just enormously informative. Just like to mention a little bit about historical controls. That did come earlier, and that's another thing that we get asked quite often. Can I use my natural history study as a historical control? Well, the answer I usually give is that depends. But natural history studies, their main purpose is to inform clinical development, but there may be infrequent times where we can use them as the control, a historical control. This is usually reserved for very special circumstances, and typically those would be diseases with high and predictable mortality and where the effect of the drug is self-evident. A frequently cited example of this, which isn't a disease but is obvious, is jumping out of an airplane without a parachute. I think that has high and predictable mortality, and the effects of the parachute, a safe landing is certainly self-evident, and to use a concurrent control in that situation, of course, would be both unnecessary and unethical, so we wouldn't do it. But situations as clear-cut as that aren't that common. So for drug development, any drug development, at the end of the day, you need to be able to interpret your results, and the whole purpose of a control is you're trying to measure what would have happened if I had not intervened. So concurrent controls are usually the strongest there. Historical controls, there's a lot of opportunity for bias to sneak in, so that's why circumstances are usually limited. I mean, just some of the factors is you will be using different patients than are being studied with the intervention. They may be on alternate treatments or may be treated differently just by the how the protocol is run, often at different times or different places or both. And you need adequate documentation. And if this is historical, that documentation may not exist. The patient populations have to be comparable, and this is a very important point. Medical practice changes. Supportive care, for example, respiratory support has changed a lot over the years. So even if you're looking back 5 to 10 years, this could have a very big impact on patient quality of life and mortality. So that those have to match up. There are two general types of historical controls. One is the informal, the implicit, based on general knowledge, or sort of the what everybody knows standard. Everybody knows if you jump out of an airplane, you're not going to do very well without a parachute. And open label single arm studies where you're comparing patients to their own baseline, that is an implied control. You're historically controlling them to what would have happened without the intervention. So it's plainly reasonable for that a dramatic effect. It's rapid after the treatment. It's very unlikely to have occurred spontaneously, and there will be a limited number of situations where we can use that. The other type of historical control is one that is specifically sought. Often, a natural history protocol will be written in advance, and then the information will be gone and retrieved. and then used specifically as a comparator in a study. That has been done sometimes. For example, L-glucosidase alpha or myozyme for infantile Pompe did use a historical control that was designed specifically for that study. It was carefully matched. There was an objective, verifiable outcome measure, in this case mortality, and it was felt to be a fair comparison to the interventional population. More subjective measures such as pain or functional measures are a lot harder to compare because there is that subjective component there that's hard to pull out retrospectively. So in terms of getting started, this is another question, is people, how do I start? How do I do this? Well, first, take a look at what already exists. There are a number of patient registries out there, and while they may not be collecting clinical data, they still can be very useful. Maybe help you find patients, it may help you find centers of excellence, and may give you some description of the disease and its prevalence and the various stages that the patients are at. Literature can be helpful, but a caution here, I wouldn't use the literature as the historical control or as a natural history study itself. What we've found as well is that literature tends to represent the more noteworthy cases. So the severe end of spectrum of the disease or something very remarkable and may not represent the disease as a whole. So it can help, it shouldn't be the whole story. Expert opinion as well is helpful, but please don't go to just one expert. People have different practices, different perceptions of what is important, and if we want to describe the whole disease, we really need to talk to multiple people. And this is a very important point, and we're surprised sometimes at how this isn't thought of or isn't done, but please talk to the patients and to the caregivers. They will tell you the important aspects of the disease, how it impacts them, and what's important to them, and patients absolutely need to be part of this process. And identifying the gaps. Again, projecting down the line, we want to design and conduct those adequate and well-controlled studies. What's missing? What are you going to need? And you need to get this in a timely fashion because we don't want to delay drug development. For example, endpoint and outcome measure assessments, they take a while to develop, need a long lead time, and we can sometimes identify and pilot those early. There are three general types of natural history studies that are used. I think a retrospective chart review is self-explanatory. Prospective longitudinal, where you're going forward, collecting data, also self-explanatory. Prospective cross-sectional, that's essentially a cross-section in time. You're assessing who the patients are and collecting and looking at a number of fields at that one point. So you could have the very young, the older patients, everybody in between, people at different stages, and you're trying to define milestones, for example. After the age of 25, everybody is on dialysis, for example. That can be very informative as you move forward. So what are the pros and cons with each of these? Retrospective is generally faster, generally cheaper, relatively speaking. The data already exist, we're collecting them. But this is also the one that is most subject to bias, and you will be limited by what's there and what somebody else thought was important, and you may or may not be able to pool it if people collected different things at different centers. Prospective longitudinal is really the gold standard. It's prospective. You're making objective assessments. You can set the fields you want to collect. Can be comprehensive, and there's a chance to learn as you go. Obvious limitations here are the time. These take a long time to conduct, and the cost. Tend to suffer from scope creep. We want to collect everything, but you can't. You don't want to bring people back several times a year for a 5-hour visit and multiple procedures, so we really have to decide what we need and what we want. And then long-term follow-up can be difficult. These can go on for years. For this particular type of natural history design, we strongly suggest staying closely involved with the patient community. Please involve the patients, and preferably that they would have a leadership and steering role in these types of studies. Prospective cross-sectional, relatively faster, combines existing and new information, but it will be limited by you're taking kind of a one look. If that snapshot has the Eiffel Tower in the background and you really needed the Grand Canyon, you may have to go back and reassess. So I'd just like to give you a couple of examples from very recent history. Here are the CDER approvals in 2012 as of March 31st. There was actually one more last week, but it's a total of 8 new molecular entities, new biologics. 3 were for rare, 5 are for common, and they're all for different diseases. And looking at disease precedent again, this is fairly typical. 2 out of 3 of the rare diseases didn't have disease precedent, and 4 or 4.5 of the common ones did. Vismodegib was for metastatic and locally advanced basal cell cancer. There are drugs approved for basal cell, but not for that particular type, so that's sort of a little bit of both. So let's just take a closer look at the 2 rare disease ones that didn't have disease. This is thought to be very low prevalence. Nobody knows exactly how often it happens, but it couldn't be more than a couple of hundred to a couple of thousand in the US every year. In the medical officer's review, it said it wasn't feasible to prospectively study glucarpidase in a randomized placebo-controlled trial. It's an emergency situation. It occurs unpredictably. We don't know who the patients are going to be. And existing treatment, which is usually emergency dialysis, is unsatisfactory. So the results showed that 10 of the 22 patients, these patients were selected because they were the only ones that had the methotrexate levels after glucarpidase administration in a retrospective review. They met the criteria for the clinically important reduction, but all of the patients had substantial reductions, and this was sustained over 8 days. So why was this felt to be adequate for determining efficacy and safety of this product? Well, there was a lot of historical information that turned out to be very, very important. Methotrexate's been around since 1948. The higher doses are used mostly for cancers, leukemias, lymphomas, sarcomas, but lower doses it's used for a lot of things, such as rheumatoid arthritis. There's a large and very long-term clinical experience with this. It's very well described. Mechanism of action, its toxicity, everything is very well understood. And the adverse effects of the toxic levels have actually, there are excretion curves which correlate with adverse events that go back to the 1970s, and this has been replicated many, many times over the years. So out of the medical officer's reviews and the division director's decisional memos, they said, these are quotes out of the reviews, they say rapid and sustained plasma levels represents a pharmacodynamic endpoint judged to be a valid surrogate endpoint, which supported a full approval. And given the extensive data, the methotrexate excretion curves are well characterized. It was sufficient to provide a clear assessment of a treatment effect. So the historical data was considered, it was assessed, and actually carried considerable weight in leading to the approval of this product. The second example this year is ivacaftor for cystic fibrosis, which I believe was mentioned a few times earlier today. And this is indicated for cystic fibrosis, a specific mutation in cystic fibrosis, which occurs in about 5% of the population. So you've now taken a rare disease and selected an even rarer subset within a rare disease. So there's only about 1,200 patients with this mutation in the US. Efficacy was demonstrated using a much more traditional plan. You had the 2 randomized double-blind placebo-controlled trials. And the division director who made the decision said that this demonstrated a robust, clinically meaningful benefit in several aspects of CF. Lung function, a change in FEV1 was the primary endpoint, pulmonary exacerbations, and substantial weight gain. Patients gained 2 to 3 kilos over the course of 26 weeks, which is absolutely remarkable and unheard of in cystic fibrosis. So what does historical information have to do with that? Well, CF has a long-standing registry. The disease is very well described. This started in about 1960, and they've collected just massive amounts of data on survival, FEV1 decline, correlations with mortality, weight gain, just about every field you can think of, they have collected it. And all of this information went into designing the program. It's a long-term chronic disease. The clinical studies were conducted over about 6 months, and it was felt that the information here and the robust findings supported full approval, and actually a rapid approval. This was approved in 3 months. This is a slide from Preston Campbell at the CF Foundation, and what it shows, a question came up earlier, is how can patients help get people interested in clinical development for rare diseases? Well, this is one very important way. Cystic Fibrosis Foundation, they have what they call a Manhattan-like project for cystic fibrosis. They've collected a massive amount of data and it had offshoots. They've also developed basic scientific research, clinical research, and other kinds of supporting programs. And this hasn't supported just the development of one drug, but for several drugs for CF, such as pancreatic enzymes and inhaled antibiotics. So this is a way of de-risking clinical development, for really developing a very solid scientific foundation and robust information that can then be used over and over again. The natural history supports the disease. It's not tied to a particular drug or development program. So this can be recycled and used over and over again. And patients are absolutely essential to collecting this information. So just to sum up then, so the natural history data contribute to the scientific foundation upon which drug development programs can be built. This is particularly important for rare diseases because we often don't have that information. We're often starting de novo. And rational, scientifically based drug development really does require a good understanding of the disease. That's not the only thing we need, but it is one of the elements, and it really will help us out. And as I just mentioned, natural history is independent of the drug. It can be used again over and over. These really do require long lead times, though, and we often recommend to people, please go ahead, start this now. If I had my way, all rare diseases would have at least a registry, give us something to build on, and I think it would really advance drug development for many of these diseases that really don't even have research going on right now. You can start even before there's a candidate identified, and ideally, for this to be maximally beneficial and to help maximally inform clinical development and for efficiency, more rapid development, we would really like to have this information before we start designing and conducting clinical trials. Patient and disease advocates are extremely important to this process. Please involve them early and actually bring all stakeholders to the table as early and on an ongoing basis as to the extent possible.